That's all
watch
right.
it, yeah.
It doesn't have to be long, does it? I don't know how long it's going to go for.
Well, 45 minutes is the normal time, but if it's plus or minus a bit, that's all right. Students will be happy if it's shorter, so
Yeah,
if it's a
I
bit
have no
longer,
idea.
that's okay too. So.
Whatever happens, happens. I
Yeah.
guess.
Okay. If you, I mean, it should be all fine, but if something goes wrong, you can always just call me or something.
Yeah, I don't actually have your phone number, I think. What's your phone number?
It's 0423 522 611.
I'll just call and then stop so that I don't lose you. The contacts.
Did you call?
Yes.
Oh, it didn't come up. If you call for a bit longer, maybe then I can add you to mine as well.
Okay.
Just realised that if that recorded me, all the students will know my phone number now.
Oh.
Oh, there we go. That's really okay. Cool.
How do we know whether it started recording? Hopefully not.
You don't know. It usually starts a couple of minutes before. So it doesn't matter. Doesn't matter.
So they can hear everything we spoke about.
Probably. Well, we were talking pretty quietly, so it's probably only maybe a minute or so. I don't think it records that long. Okay. Also, are you doctor or April? Alright, hi everyone. So today we're having a really special guest lecture by Dr. Svetlana Boltic. So Svetlana Boltic has been working in biomedical research for quite some time now, a couple of decades I think, and at UWA, but also at the Institute for Respiratory Health, based over in the Perkins building.
Mm-hmm.
So, but in more recent times she's started working in biotech, and specifically since 2020 she started up her own biotech company called Gene S. So it's a pharmacogenomics company, so it fits really nicely into these series of lectures on pharmacogenomics. So I will move over to let Svetlana give you this talk about pharmacogenomics, and I'm going to have to run out, so sorry.
Thank you, Liz. Hello everyone. So I just thought that I would give you a little update of what GNS is, and given that we are a startup and a biotech company that is here at UWA, so that you know that we exist and what we do. And then because precision medicine is what we are passionate about, I'll tell you something about technologies that are currently in use in the pharmacogenomics, and that fits nicely with what you've learned so far. So I've tried not to have a lot of overlap with what the content that you already covered, so I will refer to it, but I won't go deep into things that were covered by Phil. So it's a little bit about GNS. So we are a WA-based company. As Liz said, we founded company in September 2020, so it's actually just our third anniversary that passed by. And we have here a lab in M block on the first floor, so you can always come and visit us and see what we do. And then we have offices at SERI, which is a small incubator for the startups, and that's where we can learn something about the business. And they also run some courses for students, so if you have any idea, you can always contact me and I can connect you with SERI, so it is always good to chase your dreams. So we really have a mission to provide accessible and trusted genetic solution to facilitate personalized therapies to improve the health of everyone. So we want to have this personalized medicine available to everyone. That is what we're really trying to do. So you know what pharmacogenomic is. It is a study to look into the relationship between someone's genes and how you respond to medications. And that information, and if we do this testing, it will really allow us to predict how the patients will respond to medications. So therefore, doctors will have more informed decision when they prescribe patients and the patients medications to patients, and they will reach, they will get better quicker and sooner and avoid all these wastage of time, of money for the medication, and prevent all this hospitalization. You wouldn't believe 30% of hospitalization is actually due to adverse adverse reactions. It is a huge problem, and the government is aware that Australia spends more than $1 billion every year on preventing adverse reactions or dealing with adverse reactions from inappropriate use of medication. So there are a couple of major milestones that had to happen in pharmacogenomics to make the testing possible. So first, of course, there was a discovery of the double helix, and then the first pharmacogenomic, the term pharmacogenetic or pharmacogenomic was used by Vogel in '59. And then we started developing or discovering different polymorphisms. The databases started to create, collating all this information. And then, you know, development of all these new methodologies, sequencing, we really started to accumulate that knowledge. And now we have more than 400 drugs that are recognized by FDA that have pharmacogenomic value, or that they depend on our genes. So there is that established relationship between genes and the drugs. And in Australia, there are only 2 drugs that are currently covered by Medicare, but we know that it is coming, so it is going to become more of the general practice and, you know, in the mainstream of the patient management. So the issue is that, believe it or not, that more than 70% of the initially prescribed drugs are inefficient. And the reason for that is in our genes. So that affects 17 million people, 17 million Australians every year. And it is not restricted to one class of medications, but really across all these different disease states and medications that are used in different diseases, that's the case. So there are different now statistics when you look at that, but the majority agree that more than 98% of us carry at least one change in our DNA that would determine how we respond to medications. So there are some new studies that claim that it's 100% of us, but let's stick to 98%. And so there is going to be 30% of people that will respond to the standard dose, standard drug prescribing, but 70% won't. So it is going to be even that standard dose is ineffective, or that it can be toxic, and then toxic with benefits, or really with no benefits, and it should be avoided totally. So when you think about it, if you don't have personalized approach, so when you go to a doctor, we are all the same. They can't really distinguish between us, so they have to use their current guidelines and the best they can. So they just prescribe the medication that, you know, the previous patient responded well, so, you know, in according to their, of course, guidelines. But they prescribe medications. Some patients will respond, some will not, and some will have adverse reactions. But if with personalized medicine, so if doctors can have that information about your genetic profile, they can stratify patients, and then every patient should benefit. Okay, so that is the idea behind it. So how it works, and I here gave the example of GNS, so because we are creating genetic test and a software to solve this problem. But idea is that the patient goes to a GP or a specialist. They prescribe, as soon as they need to prescribe medications, they send patient to a local pathology lab who would do the test, send the results within a couple of days back to a doctor, and doctor prescribe the right medication that is right for patients. At the moment, what's happening, in particular in mental health, and that is the first thing that we are tackling, is that patient would go to a doctor prescribe one medication, 70% of the patients wouldn't respond. They trial, they have a couple of months of trial, then if it doesn't work, they go back. The doctor prescribes another medication, again a couple of months of trial, it doesn't work, go back. That goes for years. And some consequences are terrible, you know, like suicide is one of the most, the higher incidence of death in young people. So it is really terrible consequences for patient and for the family as a whole. So collecting and processing sample is a crucial step in this pharmacogenomic testing. So when you think about it, your patient will go to a pathology lab, and patient can give either blood sample or a buccal swab or saliva. So from blood samples, the white blood cells are used to isolate DNA, and in buccal swab or saliva, it is actually like the cells from inside of cheek that are used for DNA extraction. And every single pharmacogenomic test starts with the DNA extraction. And in some you need a good quality of DNA. Some are that the newest technologies are less sensitive to the DNA quality. So let's look into genetic variants and their impact on drug response. So there are several types of these genetic variants, so including single nucleotide polymorphisms or SNPs. There can be insertions and deletions. So sometimes it's one or two bases, insertion, deletion, but sometimes it can be a big chunk of the gene that is missing. Also, copy number variation can influence largely the pharmacogenes, and there are also structural variants, and I will touch on that later. So all of these can impact the drug metabolism targets and leading to variations in drug response and efficacy of the drugs. So these are some of the lab techniques that are currently used. So it's PCR, microarrays, and DNA sequencing. And we are going to touch on all of them so that you see what they do, what they detect, and some of advantages or disadvantages of using those techniques. So PCR, have you actually learned about PCR previously? Some yes, some no, okay, a little bit. So it is a technique that is used to amplify certain regions of the gene. So it can be, you know, sometimes a whole gene long PCR, or you can look at just a little fraction and try to detect only one SNP. So how PCR works? So it has like it goes in cycles. So first you have a DNA. Can you actually see my? You can't. Okay, let's try with this. Do you see this? Okay. So you have a genomic DNA, and if you want to amplify a certain region, you need primers. Primers actually specifically bind at that region, and they allow polymerase to transcribe and, you know, to copy that strand of DNA. So what happens, you have to first denature DNA and then enable primer annealing to specific DNA sequence, and then polymerase actually can synthesize that, so that fragment. And then in every step, so you go through those steps, so denature, annealing, and synthesis, and it goes like that, and it doubles. So in every cycle, the amount of that product that you have is actually doubling. So from a single gene copy, you can have billion copies at the end of the sequence that are created in 30, 40 cycles. So that is how that works. So if you want to look at the variant in the specific variants, and there are a couple of ways that you can go about it, but usually if, for example, variant is at the last nucleotide in the primer is not going to bind well, and it is not going to produce any product. So you lose that product, and so you basically, because for the PCR to detect, you need to run gel electrophoresis, so you have to see the presence or the absence of the product. So if you don't have, in this case, if you have the SNP there, the primer is not going to amplify it, because polymerase will not be able to bind properly there, and it will not enable polymerase to really go and extend that product. So you will end up with no product if you have SNP. If you don't have SNP, if you don't have that polymorphism, you will end up with a product like that. And if you have SNP, you will miss the product, and then when you run on the gel, you can differentiate and you can genotype that way. So you can run the gel, and nowadays you can run capillary electrophoresis, which is a bit more sensitive but also more expensive than the gels. But that is usually, running the gel takes time. So PCR is actually the first step also in a lot of other technologies that is used in microarray sequencing. It is really that first step that starts all of these technologies. And as I said, it can be used to amplify the whole gene, or you can just amplify certain regions and then run it on a gel, or use it as an input for microarray or sequencing. So because running of the gels is not always practical and it takes time, so we developed, well, not we, but the real-time PCR was developed, and the first time those primer probes were described in 1991. So it is really a long time ago, but now it is in the mainstream pathology lab screening, clinical screenings. So what's happening, like, in PCR you have primers, gene-specific primers, but in addition to that you have a probe. And the probe binds in the middle of your product, and it has like fluorescent dye on the 5' end and the quencher on the 3' end. So what happens when they are in a close proximity, the quencher actually dims down that signal from fluorescent dye, and then when you have like a normal PCR method, so you have this denature, then annealing of the primers, the probes anneal as well to the region of interest, and then when their extension is happening, so polymerase goes, and then it hydrolyzes the probe, so it releases that fluorophore. So suddenly, because fluorophore is not in a close proximity with quencher, so it releases and you can detect. So if you take a photo at that moment, you can see how much fluorescence there is. And the fluorescence is actually related to how much product you have. So you can see over time, and as the product, more and more product you have, more and more probes are going to bind in the next round to your product in the PCR cycles. And then as they are hydrolyzed, so the synthesis is going, those fluorophores are being released and being detected. So you can see in every step how you have more and more product, and then you have those curves, and you can detect if you're if you're looking for the mRNA expression, you can see how much gene expression there is. So you can look at all of that. But in terms of if we look at the polymorphisms, we can have probes that are specific to bind the one or the second allele, so the wild type or the polymorphisms, and then by looking at those, you can see whether the patient is homozygous for one allele or another allele, or you have a heterozygous where you have one copy of the wild type and one copy that is polymorphic. So that is how you see that. So the advantages of this is that you don't have to run the gel. You really see that in real time. So you run the PCR and you get your results. You can follow that in real time as the PCR goes, and this is how you can then detect. There are a lot of advantages and disadvantages of using real-time PCR. It all seems very good. So it is fast and allows that visualization and analysis. It is really sensitive, so you need less copies of the DNA of the starting material than for the conventional PCR. And so very accurate, and therefore it is cost effective. But you can use it for the copy number variation. It is used for that. It is used for SNP analysis. It can be used, as I mentioned, for the detection of the gene expression. But also, we can multiplex as well, because if you're using different fluorophores, you can put them together, and you can detect two different signals. However, there are some interference. There are sometimes, when you look at these results, they are not really stratified very well. So you have to be very careful what you put together, how they all work, and sometimes the results are not that easy to interpret. So there are some challenges with using these technologies. So another technology that is really, microarrays are a powerful tool in biomedical research, and they have become really indispensable for high throughput multiplex analysis, and especially for DNA analysis. And the basis for all microarray processing and fabrication is really that surface modification. So it is kind of a chip, and there is a substrate, and then all of these different oligonucleotides are applied on that surface. And so for the SNP array, so you have those oligonucleotides on a little chip, and you can then hybridize the regions of interest which bind to those oligos, and then you can detect. But also, if you're looking at the polymorphisms and the DNA that you apply, some will have polymorphisms, some will not. And then the ones that have polymorphisms would bind to those oligos, the others are going to be washed away, and then the signal is going to be a little bit lower. So this is how it actually works. So you have your PCR, and you amplify all the regions of interest. So if you're looking at 1,000 different targets, you have to do well, depends. If every single one is, you know, in a different region, you will have to do 1,000 different PCRs. If they are in the same position, you might be able to do less. But you start with a PCR and then you label the probe, and then you run it. You apply that DNA, patient DNA, on the chip where you have all these different oligos, and they are like, you can have really up to 1 million, I think, different probes all put together, like all of them are on a small chip, like, you know, I don't know, 1 centimeter or something. It's heaps of different. So this is how it looks like. Every single dot here actually is a different probe. So once you do that, you hybridize them, then you have a lot of these washes to get rid of all of the DNA fragments that are not bound to that, to those oligos on the chip, and then it goes into the machine with those sensitive cameras which can distinguish all of this. And then when you have here is like a red, so it is a dim, you know, signal. You have absence of that polymorphism, and when you have a green light, if it lights up, that is a presence of the polymorphisms. So you can distinguish in that way, and then the results look like this, where you have really, like you can see homozygous, heterozygous, and then those homozygous allele 1, allele 2, and then heterozygous in the middle. So you can all align according to different genes. So the key applications and advantages of this, it is really scalable, so you can put a lot on that array. So it is fairly accurate, and you can call a lot. It is quite sensitive, and therefore it is cost effective, relatively cheap. It can identify a lot of different SNPs, so you can really put those large scales. And to a certain extent, it can detect copy number variations. You have to include some controls as well. And it was widely used with GWAS studies and, you know, in those discovery areas, but it is now really used a lot in pharmacogenomics. And if you look at that, majority of labs actually use microarrays for pharmacogenomics, and then they combine with the real-time PCR for the copy number variations and for some structural variations. So most of the labs use the combination of the two methods. Another method that is used in pharmacogenomics is mass array SNP genotyping. So it really starts again with multiplex PCR, and then you have, so it amplifies the DNA sequences across the SNP side by PCR, and then uses a single extension primer to amplify PCR product. So it ensures that the primer extends only one nucleotide, and then that can be analyzed by MALDI-TOF, so mass spectrometer. And then they can look at the they look at the conformation, or, you know, like mass, and they can detect which nucleotide was incorporated. And it is really quite accurate, and has the price advantage when you analyze a lot. So that is really quite good. The concentration of the DNA really needs to be quite well to be able to use this. So the quality of DNA needs to be high. It is really accurate, it is scalable and flexible. You can multiplex a lot. and it is, as I said, used in genotyping and mutation detection, and it is quite often used by labs in pharmacogenomics. So you've probably all heard about next generation sequencing. So that is the up and coming technologies. It is not widely available to use in pathology labs for pharmacogenomic testing for various reasons, but we are developing, actually our solution is using next generation sequencing. But it really allows you to identify those genetic variations in the entire genome, but also it can be targeted. You can choose different genes to target. You don't have to go for the whole genome sequencing, even though it goes it is now much cheaper than what it used to be, but still you have all this data that you generate and that you don't necessarily need when it comes to the clinical use. So really, that's where that targeting sequencing comes. And you can have, when you look at the next generation sequencing, you can have two different ways, which is you start with PCR. So you say like, okay, I'm interested in this gene, that gene, and the third gene, and I'm going to start with a PCRing these segments of the genome, and then I'm going to sequence. Or you start with the whole genome sequencing, you fragment your DNA, and then you pull out, you hybridize, you use the probes to pull out the fragments of interest. So those are the two approaches. And then what you do when you get those fragments, you barcode them. So you label DNA from every patient, and then you can mix them together because they are barcoded. You can mix them together and push them to the sequence at the same time. So you can sequence 20 or 100 or even more patients at the same time because of those barcodes. So, and then you send them to the sequencer, and then once that's done, then you have this analysis piece, and that's quite complex. That is one other reason why it hasn't really picked up by pathology labs. But then you have all these tools that you can differentiate all these based on the barcodes. You separate them into the different patients, and then you do analysis where you can look and you can variant call. So you analyze those reads, and with this massive parallel sequencing, next generation sequencing, so every position in the DNA is sequenced 30 or 100 times, or like many times you sequence the same region. And so you can see, and you can then call variants, and you can say, okay, in this position, in that position, I can see these variations. You determine which variations it is, and then you interpret. You can say, okay, based on all of these, not just NGS, but based on all of this, if you determine the polymorphisms that you have, you can then use the databases. And I know that you had it in a previous lecture, so those are these open databases like PHAR, and then the PHAR-GKB can tell you what these variations and polymorphisms mean, and which alleles they belong to, and then once you identify the alleles, you can then call and say, okay, so these alleles actually equals this clinical recommendation. So you can write to doctors, like, based on this patient shouldn't take this drug because it's going to be toxic, or these are the drugs that this patient is going to report to. So that's how it goes. So the key advantages of NGS is really high throughput. It is really scalable, and you can multiplex because of this barcoding. what is really biggest difference between NGS and all of the prior technologies is that you do not need prior knowledge of genetic variants. So this is really a tool for discovering new genetic variants. So if you think about it, to design PCR-based approach, to design the microarray and all these oligos that you have to know and you target specific polymorphisms. If you don't know that that polymorphism exists, you can't design for that. In here, in the NGS, you can discover because you don't know what you're going to look for. So you really can, you sequence the genes and then you can discover the new polymorphism. So that is a huge advantage. And so it is less sensitive to the amount of DNA that you put into the method, and also it is depending on the approach, whether you go for the PCR or the hybrid capture. There is a different sensitivity to the quality of the DNA. So if you're starting with the PCR approach, you need intact DNA, you need good quality DNA, particularly if you're going to go for the long range, like if you need long product of couple of thousands, you need intact DNA. If the DNA is chopped, you're not going to be able to then get those long products. Okay, so that's a huge thing. So when we look at the very important pharmacogenes, and you looked at that, so there is really just 34 genes that are the highest tier, that we have the most information about and that are really valuable. And even out of them, so if you plot just the top 22, 24, you get more than 90% of the drugs that are covered. But you've learned that, and I'm going to talk now about these advantages and disadvantages for the technologies that we went through. And as an example, I'm going to give CYP2D6, which is quite complex gene and one of the major pharmacogenes. And also another HLA locus is also quite complex. And quite often, because of its complexity, in all these microarrays, they are not even typing. They are not looking for the HLA genes. They are looking actually something that is in linkage disequilibrium. So they are looking for a marker that is outside of the HLA locus that is connected with that. And it's like, hey, I've detected this polymorphism in this other gene. And I think, well, we know that in 99.8% of the time that is connected with HLA B580101, and that is how they type, which is not ideal because you can miss things and mistype. But let's go back to the CYP2D6. So CYP2D6 is actually in that locus, so you have CYP2D6, CYP2D7, CYP2D8, and it is on a negative strand, so it goes that way, so it goes this way. When we analyze it, we flip it, but that's how they sit. All of these genes are highly homologous. They're extremely similar. There are regions of particularly CYP2D6 and CYP2D7 which are identical. And there are some polymorphisms in these regions that there is absolutely no way that you can detect with PCR or PCR-based methodologies. Absolutely none. We've tried. We looked at it. It is no way that you can develop the accurate assay using PCR. So, in addition to that, so it can really, you can have a different variants, DNA variations that you can have in CYP2D6. So you can have a simple SNPs, so you can have the polymorphisms, or you can have a small insertions and deletions, and then they equal to some star alleles. Like you have one polymorphism, and you know that it is going to be end up as a star 4, which is non-functional, easy to do. However, in addition to that simple polymorphism calling, you can have a copy number variation, which means that there is star 5, which is complete deletion of the whole gene. And or you can have gene duplication or multiplication. So particularly Scandinavians, they can have multiple copies, like more than 17 copies of the CYP2D6. And then more copies you have, it depends which copies you have. Do you have functional or non-functional copies? So that is all going to impact how CYP2D6 is going to predict your response to therapy. In addition to this copy number variations, so gene deletion or gene multiplication, you can have structural variation. So you can have the structural variation when you have majority of the gene or different portion of the CYP2D7 gene combined with CYP2D6, and you know it is like the end of the gene is CYP2D6, and everything, these different portions can be done, originate from CYP2D7, and those are called CYP2D7, CYP2D6 hybrid genes. And you can have also CYP2D6, CYP2D7 hybrid genes, where you have at least exon 1 and, you know, the beginning of the CYP2D6, and then towards the end, mostly exon 9 is actually swapped for the exon 9 of CYP2D7. Exon 9s are also quite similar between the two genes and very difficult to distinguish. But also you can have those so you have the CYP2D6 and different those, but you can also have the combination and duplication of any of those tandem arrangements. So it's quite complex, and that's why current detection methods are ineffective. The reason being is if you are looking even, and I'm just going to give this as an example, and we are looking at the sequencing, and we are looking at the two different approaches to sequencing, like PCR-based, and then through that you will see PCR-based. It doesn't really matter whether you're going to do PCR only, microarray, sequencing, PCR-based sequencing, they all have the same consequence. And then we are going to look at the hybrid capture approach, so pluses and minuses. So we all start with the genomic DNA. In the PCR stream, you have to PCR it, so you have your genes of interest that you detected, you see what the sequence is, and then you create your primers that fit that gene. You amplify it, you get that, you have the pool of all the regions of interest, you barcode them, so as I said, so you index them or barcode for every patient, you mix them, and then you sequence, and you get the sequence for those regions that you created. Perfect. On the other side, hybrid capture starts a little bit different. So it starts with the DNA, and you chop it. Straight away, you fragment your DNA into the smaller fragments. So that's why this approach is less sensitive to the quality of the DNA. So first thing you do is you chop your DNA. You then index, you barcode that DNA straight away. Then you take the probes that are biotinylated, so they have biotin-based biotin on them. You use then beads that have streptavidin on that, biotin and streptavidin binds, and you put those, you use a little magnet that you capture those beads, and all of these fragments of interest that are bound to the probes with biotin are now stuck with those beads, with streptavidin beads, and you wash everything else away. Then you pull those samples together, you sequence, and you get all of these regions of interest. However, you are not really constrained with the PCR primers. So because with the PCR, you have a certain range that you can amplify. You can't go, like a long, you can't have long reads. So there are a couple of thousands that are really restrictive. You can't go where you have to introduce more PCR reactions to cover the big regions. Here you can have probes and you pick up all of these. There is no restrictions of how many probes you can have. And then you can sequence those big regions. So now let's look into the polymorphisms and why PCR sometimes can give us trouble. So again, we start with our nice design. We have our primers. And let's assume that you have a polymorphism. The patient has a polymorphism that you didn't know. It's a new polymorphism, and it is in a primer binding site. What happens, that primer is not going to bind, and you are not going to have amplification of that product. Therefore, once you sequence, you're going to miss that gene, and you're not going to have the product. You don't know whether it is deleted, you don't have it, or it is just like you don't know. So you have allele dropouts with that approach. It is particularly different so majority of the assays that are currently on the market are Caucasian based, and that's why it is not suitable for some other, for Asians or for Africans. It is not sometimes we can miss changes because of these things, because not everything is known. And there are some new polymorphisms that we just don't know about that can impact that. On the other hand, with a hybrid capture, you can have that polymorphisms, but because of the position of the probes, and probes can actually, they are long enough that they can tolerate certain percentage of mismatches. Those polymorphisms don't impact those reads and capturing those regions. So therefore, we can still sequence despite of these unknown polymorphisms. So that is what is so good about this hybrid capture project. So we ourselves, we designed the test, and I'm not going to talk too much about it, but we have now genetic tests and also have analysis and reporting software. So majority of these softwares, when you look into the pathology labs, so you can buy different tests, and as I said, like the labs usually combine microarray and then the real-time PCR for copy number variation, and then they detect what is on this microarray. They don't really have that flexibility to change what they need in the test. And then they don't have the softwares that are good for reporting. So there are some companies that develop their own software, so they buy the reagents from one company and then they develop their softwares to report. So we recognize that is one of the reasons why the pathology labs don't uptake those tests, and it is not widely available. So that's what we've done. We developed the genetic test and the software for analysis and reporting. So back to the hybrid capture and a solution and why it is really good. So it really improves the performance of the NGS. So it keeps the allele balance, so you don't have those dropouts. It can detect the copy number variation, so you can actually detect all these changes, all the possible variation in the single test, even the structural variations. So that's another thing that pathology labs are using real-time PCR for, so they use for those structural variations, but that they can be detected in the sequencing method, and particularly hybrid capture. And so you have better coverage of the genes, you improve that redundancy, and there is no PCR artifacts, because PCR, particularly multiplexing, it can have some crossover, and it allows also for multi-ethnicity coverage, as I said. The DNA, because we can use less DNA and less the lower quality DNA, we can use different methods. It is now better for all applications, because our DNA isolation methods are improving, and the methods are becoming less sensitive to that. So, but anyway, so even if you have really bad sample, most of the time it can be used in the NGS. We can multiplex. And we can really speed up the time, so we really improved the workflow. So this is a little bit about us, and that is just that we really, through our new method, this is so that you can see what are the current players in the market, but we developed a test that we can do quicker and with a better accuracy than what's out there. And this is a slide that shows really the microarray PCR-based methods with NGS and how they compare. So microarray, there are now some that are quicker and that can be done in 3 days, but majority of the microarrays actually take a week. But PCR is very fast, and NGS we can do in 2 to 3 days. The throughput is really NGS is cool. The PCR-based, if you are introducing new targets, you have to do more PCR reactions, so which impacts the DNA input that you need, the amount of DNA that you need, but also impacts the speed that you can do and the resources. And as I said, NGS is the only one that allows the novel variants. It increases the resolution. It can detect all ethnicities and all in a single platform. And this is to show you, so if we are looking at CYP2D6 and there are non-functional or non-functional alleles or alleles that have a decreased function, and out of them only up to 50% can be detected in CYP2D6 and CYP2C9 with the current methodology, which are microarray or PCRs, while with NGS we can detect 100% of them. So that is the main difference. So there are some challenges, and I'm not going to spend time on that, but there are some challenges that we have to think about in implementation of the pharmacogenomics, and we really need that standardization. We have to think about ethical, like what people do with these, how much data they have, what they do they give it to insurance companies, and all of that you probably heard in the media, etc. All of these are concerns for the patients, and it's valid concerns that we really have to educate and think about and put all of this in place. We also have to ensure the quality, so there are internal quality controls and external, and really continuous improvement is needed to ensure the quality of the pharmacogenomic testing.